Welcome back. Welcome back to a very exciting session. If we could just throw the Google Drive. So there is a Google Drive folder, and in that folder for the session is a document, a README document, that contains all sorts of useful links and information. We're going to be updating it with more useful links and information throughout the day, so we encourage you to take a look at the Google Drive folder. Welcome, come on in. Plenty of space over on this side of the room. Make yourselves comfortable. Buckle up, because we have a fantastic couple of speakers coming up. Our next speaker is Boris Bernhardt, professor at McGill University in Montreal. And Boris, over the past several years, has been expanding, pioneering, developing tools and approaches for multimodal gradient mapping using connectivity, cortical surface, high-resolution data, really a panoply of different approaches, and has been integrating these into a very usable set of software tools. So I'm excited to learn more about them. Thank you for joining the session. Thank you for sharing your work on gradients and brain space. Take it away, Boris Bernhardt.
Thank you for the kind introduction, Daniel, and for the invitation to the organizers. It's really a pleasure to be here. And I'm going to talk about gradients of brain organization, diffusion map embedding, and brain space, which is a tool that you can use to derive these gradients and to study them. So like the other speakers before, first going to talk a little bit about concepts and theory, and then I'm going to do a brain space walkthrough. The Jupyter notebooks are also on the link, so you can also download them and then get in touch if you have some problems. Yeah, let's get started. So our goal as modern day anatomists is that we try to understand a little bit cortical spatial patterning, and I think today's session is also a testament to this. And as most of you know, is that there's like really foundational anatomical work that has primarily used postmortem studies in order to chart the cortical territory. And here, this was initially based on invasive studies, so cytoarchitecture analysis, myeloarchitecture analysis. And these studies have shown that we can observe non-homogeneous patterning across the cortex. There's variations between different brain areas. And then there was a parallel set of studies that has tried to demonstrate that across the cortex, functional properties also vary in space. So most of you are familiar with the homunculus that's shown on the top from Penfield and Jaspers. That's derived from intracranial electrical stimulation studies. Everyone knows Phineas Gage, or like foundational work with patients that had brain lesions that try to find links between different brain regions and functions. And then we can also take advantage of patient populations that, for example, undergo intracranial electrophysiological recordings as part of the pre-surgical workup in order to identify functional properties in specific cortical areas. And here really the overall assumption was always that these different functional structural properties that we can derive from postmortem work also relate to interregional variations in function. And initially in traditional neuroanatomy, this was a disconnect because we couldn't do the same studies in the same populations. So we couldn't do the postmortem studies in those individuals where we also did the in vivo studies. This has now changed. We can use brain mapping techniques such as MRI, magnetic resonance imaging, and this really gives us high-res 3D information on cortical function and microstructure, and we can then also go and look at multiple scales of brain organization by trying to understand microstructure, cortical folding, we heard about this already today, and large-scale connectivity and function. And there has been a lot of groundwork to make us able to do this that relates to image processing techniques, multimodal co-registration techniques, and the way that we can fuse data in a common reference frame to finally address structure-function questions in the same individuals in vivo. So here, this is one of these examples of MRI studies that have been done, and the Human Connectome Project dataset, or HCP dataset, is like one of the big datasets with which we have started to address structure-function problems in vivo in alive humans. And these studies have also revealed that a lot of the insights that we have gained from invasive post-mortem studies can also be replicated in vivo, for example, by looking at microstructural variations using myelin proxies. And then in parallel, we can use resting state fMRI or task-based fMRI in order to identify functional properties across different brain areas. This is just a shameless pitch about some resources that we have recently released. So some of the examples can also be tested in like those studies. And here you can have pre-processed data, microstructural data, structural connectivity data, functional connectivity data in healthy individuals at 3 Tesla and 7 Tesla. So if ever you wanted to also run gradient analysis or look at eigenmodes or things like that, these could be like 2 resources in which you can do this. And this should all be downloadable, openly accessible, and you can use it. And everything here has been run with processing pipelines and co-registration. So you can look at different structure function properties across multiple scales. When we now have all these different neuroimaging modalities available in specific subjects, but also at the level of the groups and at the level of structure, function, microstructure, and connectivity, we're obviously facing like a problem of data dimensionality, because we have lots of measures across lots of different points across the cortical mantle. And then we obviously want to simplify the way that we describe cortical spatial patterning. And as some of you may know, and we had like this question already earlier from Alex, is how do we make sense of this large scale information? Because we cannot, at least with human level intelligence, try to understand thousands of data points across multiple modalities. So what we're trying to do then is we're trying to reduce cortical dimensionality. And one of the traditional approaches here is obviously cortical arealization. So from a data science perspective, this corresponds a little bit to a clustering type approach in which we try to segregate our data distribution into different discrete subunits. So we can, for example, look at cortical areas derived from cytoarchitecture. You can see the classical approaches developed in Jülich and also like recently in the Jülich brain. And then similar approaches have also been extrapolated to in vivo data using, for example, the HCP multimodal parcellation. So these are discrete approaches where we can draw discrete boundaries between different areas. Today, we're going to talk a little bit more about an alternative approach, and also from a data science perspective, this relates more to eigenvector decomposition of high-dimensional data sets. So from what will follow now is I'm going to refer to this as the gradient mapping approaches. And this is obviously foundational work by Daniel Margulis, depicted here with a chicken in his arm, that has shown that based on resting state functional MRI data from the HCP data set, we can look at the resting state connectome, and then we can reduce the dimensionality of these connectome matrices into a set of eigenvectors, so-called gradients, that describe differentiation across the cortical mantle in terms of intrinsic functional connectivity. And this is a data-driven technique, like the other ones that we've heard about today. So this purely comes from the data. And we obtain gradients in decreasing order of information that they explain in the functional connectome. And you can see the first 3 gradients depicted here in as G1, G2, and G3. And the first functional gradient, as most of you know, depicts a differentiation between sensory and visual, or sensory motor systems, and higher order heteromodal transmodal association systems, such as the default mode network. A second gradient describes then a differentiation between peripheral systems, so visual versus somatomotor, and a third gradient describes differentiation between the multiple demand network and the rest of the brain. So we have a taxonomy with which we can then understand differences in functional organization from a purely data-driven perspective. And what was really interesting from this work is, on the one hand, what you can see here. I hope the laser pointer works. Yeah. So on the one hand here, despite being derived from in vivo resting state data in a data-driven manner, this framework recapitulated foundational models of cortical hierarchical processing with a more sensory to association axis depicted as gradient 1, and this more peripheral systems depicted in gradient 2. That it relates to cortical hierarchy was also confirmed by cross-referencing the data-driven gradients with neurosynth-derived meta-analysis maps. So we can see that it recapitulates cortical cognitive hierarchies. So going from these more sensory processes towards, indeed, these higher order processes. And the third really foundational insight that came from this work is also that we have a way to represent the whole connectome as a map. So this is very useful because then we can directly look at structure-function correspondence. And here in this work, Daniel and colleagues, they have shown that the first principal gradient also relates to distance between the sensory and the higher order default mode network regions. So this gives us a really useful framework to look at whole brain organizational patterns. This is maybe just like a more semi-defensive slide in which we are kind of trying to really say what we can do with gradient mapping techniques and what we cannot do and what the assumptions are. So on the one hand, we can derive gradients from parcellated or from non-parcellated data. So that I think this is important because there has also been some debate in the community about what these gradient mapping techniques are. So the initial substrate from which we can derive the gradient is irrelevant. We can basically use the Schaefer parcellation. We can use the Glasser parcellation. We can use other type of parcellations, still derive gradients in order to obtain information on interregional or intersystems topographies. We can also derive these gradients at the level of specific brain regions. So for example, we can use an insular mask here. This is work done by Jessica from my lab, and then derive these gradients of microstructural differentiation, for example, within a specific structure. So we can also look at subregional heterogeneity with respect to different gradients. Then on the other hand, I mean, this is like implicit in here, so we can derive the gradients not only from resting state fMRI data or from task fMRI data, but we can also use other properties of interregional similarity from which we can then derive the gradients. Here in this case, for example, microstructure derived from T1 over T2 ratio mapping, or from big brain or other proxies of intracortical microstructure. And then this is maybe like another side note of this is that many folks are focused on the first gradient because it's so appealing and it's so closely related to cortical hierarchy, but the embedding actually gives us multiple components. So we can describe cortical functional organization across multiple components. And despite these gradients sometimes appearing as smooth, when we collectively analyze different gradients, we can actually obtain a really high degree of functional specificity at specific cortical points, because this is the combined effect of overlapping gradients. A lot of enthusiasm has been generated by using these low-dimensional representations, so from gradient, but also from other techniques. So this year's work from Daniel again and Julia Hundenburg, and where the gradient-based dimensions have been proposed to serve as intrinsic coordinate systems of the brain, so-called brain spaces, where we can arrange different cortical regions relative to gradients, but maybe also to other features of cortical organization. Here, this beautiful work from Ted and from Valerie Sitnor showing that these gradients or the sensory association axis indeed also not only reflects cortical hierarchies, but also spatial-temporal trajectories in cortical development, things like evolution, allometric scaling, other properties of the brain. So by having this low-dimensional coordinate system, we can maybe more easily understand overall processes that happen in the brain across short time spans, across evolutionary time spans, but maybe also across development, to ultimately then also try to understand things like plasticity, inter-individual variation, and susceptibility to psychopathology. And this is relatively easy to do now. Just a few lines of code are needed in order to derive these gradients. And today I'm going to talk primarily about BrainSpace, which is the toolbox that was developed by Reinder and by Walid, so a PhD student and postdoc in my lab back in the days. And this was like a post-OHBM trainee-driven project, really, so really nice. You know, a lot of people have been doing gradient work a few years ago at OHBM, and then, you know, the trainees basically said, hey, let's write a toolbox. So it didn't take so long, and at the end it was kind of one of the most successful things that my lab has done so far. So really good. So in a nutshell, like how these techniques work is basically we get a functional connectivity matrix, can also be a microstructural covariance matrix or something else. Then we compute, or then we apply an affinity kernel to it. So for example, cosine similarity. So this is a second order distance map of connectivity, if you want. It helps us to get a positive definite and square matrix, and from that we can then derive components using, for example, principal component analysis, Laplacian eigenmaps, or diffusion maps. And these we can then display on the cortical surface. And we can then again use, for example, specific components to resort our connectivity matrix. So here you can see the same matrix resorted along the first gradient. So you can really see this really nice spatial, or this really nice structured pattern. So the different types of dimensionality reduction that are implemented in brain space, in a nutshell, they're here. So here we implement PCA, so this is basically just an eigendecomposition of the data, Laplacian eigenmaps. We heard about the Laplacian matrix already before, and with the diffusion map, we basically apply a diffusion operator before to that same data in order to get more towards like a multi-scale process. So not just local neighborhoods between different points, but we can actually vary diffusion times so to understand maybe even more polysynaptic relationships between different data points. So this is a little bit how it looks like. I hope it's correct because I did it in the plane. So say we use more nonlinear techniques such as diffusion maps in order to be able to resolve more complex topologies in the data. So here this is like a typical nonlinear data cloud. So, and then we have a lot of points, and what we can do is we can calculate the pairwise distances between the points. Then we can run this through a kernel and also prioritize very close distances, for example, so that we basically focus more on the local neighborhoods between them. And then we apply this diffusion operator, which is in a way equivalent to having like a random walker in the graph, and like nodes that are very similar. The walker is more frequent to visit as it walks through our network. And then we can basically resolve this complex topology, so we get this manifold from the data, which would then be equivalent to the first eigenvector. And for diffusion map embedding, so there's obviously like multiple nonlinear dimensionality reduction techniques, but here we basically, as was shown in this pseudo formula that I've shown before, we get the eigenvectors really from this diffusion operator applied to the data. And by also being able to vary the diffusion times, we can integrate these more local properties of the graph, but also these more global properties. And overall, it's relatively robust to noise and computationally cheap, so it's a tractable technique. I think there was like the question already before, so we can run these diffusion maps on like 10,000 points. One has to wait a little while, but at one point you can also have it. This is just what I've already shown before. So here we have the graph. We can then convert it into this transition probability map. And basically for brain space or for like the diffusion maps, we have like these two parameters. One of them is the alpha parameter that's a little bit, weighting the density of the points. And usually we have as a default 0.5, which is a little bit in between. And then we can also vary the diffusion time, so we basically can also go towards these more polysynaptic connections in the graph. So we can kind of play with these two parameters in order to also emphasize maybe local sharp differences versus these more global and smooth topologies. I mean, this is like an ongoing thing. I think the initial key paper has used diffusion map embedding, and a lot of folks have kind of also done it, and like in my group we're also doing this, but obviously there's like alternative approaches, so sometimes the value of whether one has to use diffusion map embedding can be questioned for like some of the data. It's at least a technique that's there and that can explain additional information sometimes in more nonlinear cases where PCA may be challenged. But it also comes at a cost, interpretability of like more nonlinear techniques compared to these more linear ones. We have to tune the two parameters that I've just mentioned, so the alpha and the diffusion time. So these can sometimes lead to different results. Sometimes they can also lead to the same results. Computation cost is overall higher when we use these more nonlinear versions. Robustness to noise, it's a bit of a question. I think generally like PCA is quite robust as well. And then I think where these more nonlinear methods are overall challenge is that sometimes if we want to concatenate the projections across like, out of sample projection, for example, then it becomes like a little bit more tricky. I think when you consider like looking at the first few eigenvectors, overall results converge at least with the kind of data that we're looking at usually when we have, for example, parcellated data and when we do things at the population level. So this is work by Suk-Jun Hong. I encourage you to look at this paper where extensive validations were done with respect to test-retest reliability, prediction of phenotypes based on different settings of these decomposition techniques. I think, yeah, we were encouraged to have like these questions, right? So the first one here is, so diffusion map embedding is a linear decomposition technique. True or false? True? False? Great. The second one, longer diffusion times in the diffusion map embedding approach, they resolve more global, smoother gradients. True? Yeah, true. And then the third one, gradients can be derived from functional data only. Is that true? Is it false? Thank you. I think we have like a little bit of time. So now I'm going to talk about the gradient, about the brain space walkthrough to be like a little bit more practical. If you want to code along, you can download the Jupyter notebooks from the website. I'm also not going to risk it, so I'm going to give you like a walkthrough on the slides. I think this is safer, right? So, but the toolbox, you can download it here. So it's all on GitHub. You can pick your poison basically. So MATLAB or Python. So the boomers here, they are like maybe more on this side. And the cool ones, they will choose this one, right? Okay. And the paper is here, so have a look. And so the tutorials, they are also all on this website. I changed like a few little things because the visualization was like a little bit iffy. So maybe, you know, like if you have problems with the visualizations from the tutorial on your computer, then go with the examples that I have here. You just have to add like a few parameters to the functions. So first you have to install the thing. So here, pip install brainspace, and then you also may have to install some dependencies. And then MATLAB, you just have to download the thing from the GitHub and set your path, and it should work. So when we start doing this, so this is all from the tutorial. So here, the way it works is we basically load a bunch of stuff, and then we basically load the data that we play with, and as well as the surfaces, because we need this for display. So here I'm basically just showing this connectivity matrix on which we'll do like most of the walkthrough, right? So this is like a typical Schaefer 400 resting state functional connectivity matrix. And then we can plot the data. So here we're basically plotting the parcellation. And these are like the new things that I've added to make your visualization maybe more robust. So, and then you get the first gradients. So it's basically a few lines of code, and you're like at the gradient level. So here basically you build a gradient map object. Here you query it to derive 10 components. The rest will all be default parameters. So diffusion map embedding is set as default. Normalized angle is set as affinity matrix default, no alignment. And you fit it to your matrix, and then you get your two, you can plot your two first gradients, and you're done. So, and then you correlate this with something, and your paper is basically written, right? But don't forget about the spin tests. And then for like these different gradients, you can obviously also plot the eigenvalues for them. So here you have the scree plot with the eigenvalue decay. You can see that the first two have relatively high eigenvalues compared to the others. So, I mean, you can look at all of them, but like in this case, it's justified to only look at the first two. So this is basically just like a bit of customization. So, you know, you can also specify the different kernels. So here you run with different types of kernels. You loop over the same function that I've shown you before. So basically, you can see how with like different kernels, you can get also slightly different results when you do your analysis. In addition, there's like also like an option that you can set the sparsity of like what you want to consider. By default, it's 0.9. So you have a 10% sparsity. This is row-wise sparsity of the matrix, but nothing stops you from also maybe looking at more data points. And here, this then is the different embeddings that you can run. So here you can see the PCA, Laplacian eigenmaps, diffusion maps. You can see that they all resolve you things kind of the same way. I think we had this already, James was also answering this question, so the units don't really mean anything, right? Like, and also the directionality doesn't mean anything in this case. You need to have it relative to like some kind of template aligned, and then you can maybe start doing some comparisons. Yeah, and here, these are just the way that alignments work. So we can load a different connectivity matrix. So this is just here holdout. It's just a different group. And then we can kind of compare the different gradients from the different groups. They look relatively similar. So the example is maybe not so well chosen. But after alignment, they look a little bit closer, basically. So when you do like an alignment, they look a little bit closer. Here's like a little bit the thing that we had in the paper. So these alignments, they can then sometimes also deal with these trivial sign flips across different subjects, but maybe also across different modalities. And obviously, overall, the premise of doing alignment of the data is that things become more similar. And we're then also more able to compare apples with apples and oranges with oranges when we do between group analysis or when we look at individual variations with respect to certain gradients. So if you want to do this, make sure that you have an alignment carried out. And yeah, generally for like the gradient mapping techniques, we basically use a Procrustes rotation. So we basically just make the components more similar. Yeah, so this is basically like also one of the examples that's like on the website. So here we can also look at things that are like maybe a little bit more funky, like gradient fusion, or we can look at structure function gradients. So we can basically plot this functional connectome, and then we get this MPC connectome. This is basically microstructure profile covariance in a different parcellation here. So here, these are just the seed-based maps. So we can look at the resting state functional connectivity, but also maybe at this microstructure similarity of different brain regions. And then we can concatenate these matrices. So here we do like a vertical concatenation of them. And then we can derive the gradients from them. Where's this? It's here. So then we can derive the gradients from them. And here we get like this joint structure function gradient. So you can do kind of different things. So you can derive the gradients independently for the different modalities and then compare them, or you can derive a gradient from two modalities at the same time. So yeah, some strengths of the approach. So they map cortical spatial patterning and structure function relationships, because we really can we don't only have to look at the matrix, but we can really decompose the data into a map, and then we can relate this map obviously between different modes, different components, but also to other more regional features of cortical organization, like cortical thickness, for example. And yeah, I think by now a lot of people have done, looking at these gradients, how they are preserved across species, how they change in development, but also maybe how they are consistent, but maybe also diverging across modalities, to really see that there is something that really reflects underlying anatomy. And BrainSpace, so this is the tool that we have. There's others as well. So this is really a gradients go-to toolbox. So it should be relatively easy to use. If you have some problems, just write like a GitHub issue, and then we'll try to help you. And it's available in MATLAB and Python. So you can really be hopefully flexible with how you want to do the analysis. Yeah, we have a couple of options there. So I mentioned the Laplacian eigenmaps, the diffusion maps, the PCA, different types of alignment. There's also some null models incorporated if you ever want to compare your gradients to other modalities. I've not covered this here, but there's like Morin's spectral randomization, the spin test, and brain smash in it, I think. Eigenstrapping, I'm not sure. Maybe. Yeah, and it's usually just a few lines of code, and you should get your gradients. And it's fairly well documented and version controlled. So, like when you run it, it should work. So a few caveats. So I think this is basically, there's no real firm standards on like which kernel to use. And these all have like an impact on the results. So the choice of the embedding, obviously, also may change results. So often, when you want to just verify whether your results are consistent in your studies, try to just replicate it maybe with like other parameter variations. I think this is like something that is like a little bit, this is like worrying me sometimes, is that the way that we align the data also has an impact on the inference when we look at inter-individual variations. And some of you may be old enough to also know about like a technique that was called VBM. You know, like in the 90s and in the 2000s, people used this technique a lot. But there was also like a big debate about it because of this close connection between registration quality and inference. So here we have something that's a little bit similar. Like the better we align the gradients, the less we find. So, or the more we find it. It's like, so if somebody has like a solution for this, please help us. And I think this is like maybe sometimes when you work on gradient-based analysis and you compare your groups and you find patterns that are maybe statistically significant, the findings that you have, they're not always purely local, right? So because they also always reflect the embedding of a region with respect to the entire connectome. So we always have to see maybe what are the local properties that we could isolate within specific brain regions. And then I think this is basically that, you know, obviously there has been like a big focus on like gradient 1, but yeah, the other ones are equally important and they may all be differentially sensitive to different aspects of the brain. Chateau of possibilities. So I think, yeah, we had like the gradient workshop yesterday. Some of you were there. And I think this is basically next year. It's going to be in, where is it going to be? It's going to be at the Chateau of Possibilities. So if you wanna join the gradient workshop next year, where a lot of like-minded folks come together and talk about gradients, also a little bit about parcellations, right? I think everyone is welcome. So please, I think it's on the 12th. 12th to 13. Yeah, of June. So thank you.
Please stay by the microphone. Are there questions? I'm sure there are questions. Thank you, Boris, for a wonderful talk.
Thank you.
Yes,
oh, very excited. I actually have two questions. It's a bit personal because we work on connect topic maps. So the first question is I wanna hear your, maybe it's for Daniel as well. I wanna hear your response to the overly discussed question about the intrinsic properties of fMRI data acquisition about the case space and reconstruction via reverse Fourier transformation, for example. And then the gradient, global gradients, to be specific, is smooth, while in the recent review from Steve Peterson also mentioned that it is biologically doubtful, as you know, local sharp changes of, for example, cycle architecture, cell types, receptor types, etc. I want to hear from you guys.
Should I take? Yeah. Do you wanna answer? Okay. Yeah, I think a bit like as I mentioned also at the beginning of the talk is that high-dimensional data sets can be analyzed with different approaches. So, and we heard this already before that, you know, all models are wrong, but some are like maybe more useful. So I guess it's the same thing as in other domains that sometimes you advocate maybe for a clustering approach and this can give you certain good insights. But if you have like more of an eigenvector decomposition type approach, you have other insights that might be alternative. So it's a little bit like describing, you know, light in terms of waves and particles, right? That you can kind of look at complementary ways to look at the underlying structure of your data. Spatial autocorrelation, as it comes from acquisition, obviously it's always something that we have to be mindful of. Sometimes the image processing may also increase spatial autocorrelation. Everyone knows that you might smooth data, you might interpolate data when you co-register subjects. But some of these gradients have also been derived from, you know, cytoarchitecture data, and the findings are largely consistent. So overall, it's not just a methodological artifact. And as I said at the beginning, the gradient approaches by themselves are relatively agnostic to what the input data is. It can be parcellated data. It can be vertex-wise data. It can be voxel data. So I don't see necessarily personally the the dissonance between these different approaches. I think they should rather go hand in hand.
Okay.
In contrast to like how that paper was written. So.
Good to know. The second question is, what do you think of the intrinsic number of eigenmodes of the brain? Is it like 3, 30, 300?
I think it depends on what you want to show. I think the first few gradients, at least when we look at resting state functional connectivity, they seem to be like rather, systematic principles of brain organization, such as sensory to transmodal association, and also reflective of maybe larger scale developmental patterns. Gradient 3, for example, is maybe more useful in older age when you look at cognitive decline, and then maybe like more higher order gradients may be able to better resolve inter-individual variation at the level of, phenotypic prediction. So it's really problem dependent.
Yeah. I'm a little bit worried about, it turned out to be a result-orientated problem solving, but when you consider the fundamental, let's say our three-dimensional space and whether our brain was developed under chemical gradients, for example, and there's only 3 possible dimensions to grow your axons and dendrites in. So, yeah, it's remain discovering. Thank you.
Do you also want to? No?
Hi. I wonder whether there are any pitfalls or recommendations on how to integrate cortical, subcortical brainstem and cerebellar gradients. So, for example, scaling them together or separately. Is there any way to go?
It's also a fantastic question, right? Because sometimes when we, for example, want to derive whole brain gradients, but we represent each subcortical structure just with one number, but we have thousands of points on the cortex, then obviously the subcortical regions are not so well represented. And on the other hand, if we take parcellated data at the level of the cortex, but take all the voxels in subcortical regions, then the connectome and like the resulting embedding will be like heavily influenced by the voxels that we have. So I think one needs to kind of find like a fair compromise between both regions. But I think there's like even like a larger problem of like integrating subcortical and cortical regions, for example, with respect to SNR that we have from the coils, a lot of SNR decay towards subcortical structures. So yeah, the perfect approach I don't think has been developed yet, but there's like a pretty cool parcellation by Yitian that also combines gradient and boundary drawing approaches in subcortical structures. Daniel is like also a co-author on this. I think that's like Nature Neuroscience from 2020. So this could be like a cool approach.
So did I get it right that that you would have a slight tendency for doing it separately, the subcortical and cortical gradients and results, and then maybe combining them in the next step?
Yeah, it could be.
Thank you.
Yeah.
Thank you very much, Professor. That I have a small question that because we know when we apply to use a gradient application that we always generated the first gradient and the second gradient, and they also have the relatively meaningful. So now that there are some studies use the third and gradient, but how does the meaning of the third gradient? And I found that the paper says the meaning of the third gradient may be controversial. So can I consult with that?
The way I commonly interpret it, or like I think this is like also like relatively similar to how Daniel and Johnny Smallwood, for example, conceptualized the gradient, is that it's like a multiple demand network differentiation from the rest of the brain.
Okay, thank you very much.
We have time for one more quick question. If there are other questions, I'm just going to pull one from online that seems quite relevant. You already touched on this, Boris, but with all these parameters to generate gradients, how do we determine the best combination to identify the ground truth? So there are a couple of charged elements of that, but what to do in the face of all these parameters?
Yeah, as I said before, ideally you try to vary a bit the parameter space and then you see whether your results are robust. I don't think we have a ground truth at this point.
Thank you so much. Please join me in thanking